All right, good morning, everyone. Welcome to another iteration of the ASBMB Lipid Research Division talks. We're coming back after our summer hiatus, so we're going to be back on a once-a-month format. So as a reminder, we're doing a slightly altered schedule for the next 2 months. We're going to be the first week of November and the first week of December for our next two talks. The only other big piece of news is that currently the ASBMB annual meeting registration is now open. There's going to be an LRD division talk at that meeting. And also the co-organizer of this session, or co-organizer of this meeting setup, Mike Arola, is going to win a big award there. So please come and check that out in person. It'll be great to actually see a lot of people's faces back in person. All right, with that, we'll get started with the meetings. As a reminder, please type in your questions in the chat box, and you'll be called upon to ask your questions at the end of the talk. And with that, I'm excited to introduce the first speaker and introduce the introducer of the first speaker. So the first speaker is Nivea Pereira de Sa, and it's my pleasure to introduce Dr. Maurizio Del Poeta to introduce her. Take it away.
Nivea Pereira de Sa graduated in biological science at the Federal University of Minas Gerais in 2010. From the same university, she obtained a master program in 2013 and a PhD in 2017. Her PhD work consisted in testing new antifungal compounds against a variety of fungal organisms using in vitro and in vivo models of fungal infection. Her advisor at that time for a master program was Susanna Joanne and Patricia Silvia Ciusalpino. For her PhD, she was co-mentored by Susanna Joanne and Daniel de Assis Santos. During her PhD 2015, Nivea did a research fellowship in the laboratory of Dr. Eleftherios Milonakis, chief of the Division of Infection Diseases at Brown University, where she studied the antifungal activity of new compounds using the invertebrate model of infection. Nivea joined my lab in March 2018, and since then she has been working on 3 drug development projects. The work that she will present today on the sterylglucosidase 1, or SGL1, is her main project, which was done in collaboration with Mike Airola from the Department of Biochemistry at Stony Brook University. His work has been just accepted for publication. Nivea not only was able to solve the crystal structure of this fungal enzyme, but she was also able to set up a high-throughput screening assay to test the Cambridge diverse library and identify first-class inhibitors of SGL1. Congratulations, Nivea, on this accomplishment. Mike and I are so proud of the scientists you have grown to be and will continue to watch you grow. Looking forward to see what else you achieve as we move forward. So, Nivea.
Thank you, Maurizio, and thank you for the opportunity to be presenting my work here today. The title of my talk is Structure and Inhibition of Cryptococcus neoformans sterylglucosidase to Develop Antifungal Agents. Well, fungal infections are a really major problem in hospital settings, and they primarily affect immunocompromised people, and the treatments are lacking. The mortality rate are over 40 to 60%, and we haven't had any new therapy for deadly pathogens approved by the FDA in over 20 years. So my work is like showing how targeting a glycolipid metabolism would lead to new therapies. And this is the story I'm gonna tell you now. So human cryptococcus opportunistic in a primary respiratory fungal infection that it's caused by Cryptococcus. There are mainly 2 species, Cryptococcus gattii, that it's frequently affect immunocompetent people, and Cryptococcus neoformans that affect immunocompromised individuals. But independent of the species, the infection happened by the inhalation of infectious propagules that are present in the environment. And once it reached the lungs, it developed to a pulmonary disease and then disseminate to the brain, causing meningitis. And because the immunocompromised population is highly susceptible to these infections, the HIV population is highly affected with over 200,000 cases of cryptococcal meningitis every year. And the anti-cryptococcal therapy we have available counts basically on 3 drugs: fluconazole, amphotericin B, and flucytosine. But fluconazole, its use is limited by its drug interactions and emergency of resistant strains. Amphotericin B major drawback is the high toxicity to the kidneys, and flucytosine that's also toxic. It's only available in a few countries. So it's a critical need to develop pharmacological agents. And our group is studying a special kind of glycolipids called the sterylglucosides. These lipids are present in many different organisms, but in each one with unique structures. Here are 3 examples of the structures of these lipids. In fungi, the function is quite enigmatic, and we know that they are present in very low levels in the cells. And we also know that if we inject them in animals, it stimulates immune response. We also know the proteins that are involved in the metabolism of sterylglucosides, but they have not been characterized yet. So in the fungal sterylglucosides is formed by a sterylglucosyl transferase that add a glucose moiety to the 3-beta-hydroxy group of a free ergosterol, forming the ergosterol glucoside that is then break down by a sterylglucosidase. And sterylglucosidase from Cryptococcus neoformans SGL1 is the first enzyme to be identified with sterylglucosidase activity. And it's interesting that this enzyme is universally conserved in fungi, not found in humans. So it's interesting that when we delete SGL1 gene from Cryptococcus, it renders the mutant non-pathogenic. So here I'm showing a model of inhalation model of infection that mimics what happens in humans, where the yeast is inhaled, cause a pulmonary disease, and then disseminate to the brain. And here in this survival experiment, they infected 3 groups of mice, one with the Cryptococcus wild type, a second group with a mutant lacking SGL1, and a third group with the reconstituted strain. And what they see is that there is 100% survival when infected with the mutant delta SGL1, while all the other animals that were infected with wild type and reconstituted die until 30 days. And if we look at the lungs, we see that the mutant start dying and the CFU number in the lungs decrease until 14 days, the lungs is completely cleared. And this doesn't happen when we infect with wild type or reconstituted. And the most interesting yet is that at the brain with wild type and reconstituted, we can already isolate yeast from the brain after 6 days of infection. And the mutant, it's never disseminating to the brain. So this data suggests that SGL1 is a promising therapeutic target for cryptococcosis, and as it is universally conserved, potentially also for other fungal infections. So our goals was basically understand the Cryptococcus SGL1 substrate specificity and identify anti-SGL1 compounds. And the primary question we have was about the substrate specificity. Watanabe had originally proposed this enzyme as a glucosylceramidase, and based mainly in the activity against the non-physiological substrate C6-NBD glucosylceramide. So I expressed this protein SGL1 in nuclei and purify it, and then I assessed the enzymatic activity, confirming what Watanabe has mentioned before. But when we look at the activity, compare this with the activity against the ergosterol glucoside, the activity for C6 glucosylceramide, it's almost negligible. And the activity for ergosterol glucoside is much higher. And different from that paper, we also tested the activity of SGL1 against the endogenous glucosylceramide that's present in Cryptococcus. And we were not able to detect any activity using this substrate, while the human glucosylceramidase, it's readily able to do it. So this confirms SGL1 as a sterylglucosidase. And to understand better the substrate specificity, we thought in determining the structure of SGL1. And initially, the full-length protein failed to crystallize. So we identified a major region, disorder region in this protein, and we delete 55 residues from it. From a region that doesn't seem to be conserved among other SGL1 orthologs. And then we got the structure that, as you can see here in figure B, SGL1 has basically two domains. One large catalytic domain with a central tin barrel and a C-terminal beta sandwich domain. And what is interesting is that SGL1 has this large unique region here that forms a cap. And this cap basically encloses the active site, forming an Y-shaped cavity that apparently limits the binding to just one single glucose moiety here. And in the arms of this Y-shaped, they are lined up with hydrophobic residues, and one of the arms is completely buried inside the protein by the cap, and the other arm, it has a narrow opening that is solvent exposed. And this structure of SGL1, it resembles a lot the structure of a bacterial glucosylceramidase 2 and the human glucosylceramidase, but differing in many significant ways. The bacterial enzyme doesn't have that huge cap enclosing the active site. Here, the active site is completely open, and it allows large lipids to bind with multiple sugars. In the human enzyme, there is a cap, but it's much smaller than the SGL1 cap, and still the active site is completely solvent exposed. So the question that still remained was, what really makes SGL1 active site unable to feed the endogenous glucosylceramide? And we were unable to co-crystallize the SGL1 with ergosteroglucoside. So we use a computational approach to dock the substrate. And this model showed that there are two glutamates, two catalytic glutamates in a canonical topological position for retaining beta-glucosidase activities. And if we look at the residues around surrounding the glucose, they are very similar to the residues present in the bacterial enzyme and also in the human enzymes. But if we compare the capsules that are in the SGL1 and in the human enzyme, the residues in this cap for SGL1 are very different from the residues in the human enzyme, what suggests that a selective SGL1 inhibitor wouldn't inhibit the human enzyme. So also use the same protocol to dock the C6 glucosylceramide and the fungal glucosylceramide. And what we can see here in yellow is that the glucose of C6 glucosylceramide is in an identical position as the glucose of ergosteroglucoside. It's consistent with the activity we have seen in vitro. But if we look here in green, the fungal glucosylceramide, it's in a kind of contort, and the glucose is in a position that wouldn't allow hydrolysis. So this shows that SGL1 generates the substrate specificity for ergosteroglucoside through its enclosed Y-shaped cavity. And after this, as SGL1 is a promising therapeutic target and exclusive from fungal cells, we develop a high-throughput screening assay using a resorphin glucopyranoside as a substrate, and I screened over 50,000 compounds. From this library, I identified 15 hits anti-SGL1 And then I evaluate the toxicity from these hits, and also the ability to lead to an accumulation of ergosteroglucoside in Cryptococcus cells. And I end up with 3 hits. From these 3 hits, hit 1, it's a thiazole derivative, and hit 9 and 15 are from the same chemical class. They share this methylpyrrolidine ring, and the activities, the IC50 for these 3 inhibitors are pretty similar, 0.5 to 1 micromolar. And the toxicity for mammalian cells, the LD50 is higher than 500 or 600 micromolar. So the selectivity index for these compounds are very high. So as I have done the screening with a different substrate, I tested it again using the natural substrate, ergosteroglucoside, and I could see dose-dependent inhibition using this substrate. And then I treated Cryptococcus Y-type cells with these 3 hits. And I could see also a dose-dependent accumulation of ergosteroglucoside inside the Y-type cells, which is significant when compared with the Y-type cells without any treatment. But, well, these compounds are blocking the activity of SGL1 in vitro, which is good, but still the accumulation of ergosteroglucoside in crypto cells are about 20% of the accumulation we usually see when we mutate the gene. And so before I move forward with these compounds, we also thought and developed a secondary cell-based assay to screen compounds against SGL1. And delta SGL1 mutant doesn't have a clear phenotype. But when we seal the plates and then incubate them, we can see that only the Y-type and the reconstituted strains are able to grow. And delta SGL1 can't grow in this low or in this condition of oxygen deprivation. And it's interesting that treating the Y-type with these 3 hits, hit 1, 9, and 15, we can reproduce this growth defect. And this is quite intriguing because it's very similar to the condition, the oxygen condition that we see in the host environment. So remember, the infection happened by the inhalation of the yeast, it reaches the lung, and inside the alveolar space, the oxygen concentration is very high. But once the yeast is in this environment, the macrophage can try to clear it out. And intracellularly, the oxygen concentration is much lower. And if it moves to the interstitial space, the concentration is also lower. And even at the brain, the concentration is much lower than the alveolar space. So maybe that's why the mutant delta SGL1 is cleared in vivo. So SGL1 are very promising, inhibitors are very promising from a therapeutic perspective. So what we want to do is to test these hits in vivo. And we have already started this with hit 1. And I have done a model of inhalation, of model of infection, the same way that inhaling this yeast, it causes pulmonary disease and then disseminates to the brain. So I follow the lung burden, fungal burden during 14 days. And I treat these animals with 10 milligram per kilo per day of hit 1 or fluconazole, or hit 1 in combination with fluconazole. And after 3 days of infection, there was no significant difference among these groups, treated or not. After 6 days, again, no difference. And also no difference after 9 days of treatment. With 14 days, we start to see some difference, especially with the combination of hit 1 and fluconazole. But what was more interesting was that every time I have hit one, there was no brain dissemination. So I could only isolate crypto cells from fluconazole treatment or the control group. And this is interesting because even though this compound only leads to an accumulation of ergosterolglucoside of about 20% of what we see in the mutant, it still was able to cause this great effect in vivo with no brain dissemination. So optimize these compounds and improve the potency of them seem to be the key to increase this accumulation of ergosterolglucoside and possibly leading to a clearance in vivo. So to optimize these compounds, to do that, I co-crystallized the SGL1 with HIT1 and HIT9. And what we see is that both compounds are binding exactly in the glucose binding site. And with this information, we are now able to start optimizing these first-in-class SGL1 inhibitors using traditional medicinal chemistry or computational approach. And so what I just presented was that Cryptococcus neoformans SGL1 acts only as a sterylglucosidase specifically. And we have revealed the structural basis for the substrate specificity. And we identified the first-in-class inhibitors anti-SGL1 that lead to an accumulation of ergosterolglucoside and Cryptococcus Y-type cells. And when Y-type cells are treated with these compounds, they mimic the growth defect that we saw in the mutant. And the treatment with HIT1 decreases the virulence of Y-type Cryptococcus neoformans in the mouse model of infection. So these compounds are the first chemical tools to manipulate the ergosterol levels in fungi and provides then a starting point for the optimization for a medicinal chemistry study of these compounds. So I would like to thank all my co-authors in this paper, Adam, Dr. Jinhu, Dr. Timothy, Dr. Reese, Dr. John, Dr. Robert, Dr. Regina, Dr. Maurizio, and Dr. Michael Rollo. And also I would like to thank all the Poeta Lab members and all Arola Lab members and all Dr. Jima's lab members. So thank you all for being here today. Thank you for your attention.
Great, Nivea, thank you very much for a really great presentation. We've got our first questions coming in here already, so I'll go straight to those. The first question is from Jeremy Thorner. Jeremy, you should have the capability to unmute yourself and ask the question.
Okay, very nice data, nice talk, very clear. But one thing you didn't mention is, where is SGL1 localized? Is it a secreted enzyme? Is it an intracellular enzyme? Is it a membrane, peripheral membrane protein? What's known about that? And the other thing is, why do they make this glucosylated ergosterol in the first place? What one could argue that it's sort of a counteracting pathway to preventing fatty acylation of ergosterol, because some of the sterol, of course, gets stored in lipid droplets as sterol esters. So is this a way to sort of regulate the pool that's available to go into lipid droplets? What's known about why they glucosylate the ergosterol in the first place? So those are the two questions.
All right. Well, Jeremy, this enzyme SGL1, it's intracellularly. It's not a membrane enzyme. And this about the ergosterolglucoside, this is not clear yet. We don't have a clear answer for what is the function of this lipid in the cell. It could be the hypothesis that you said, but it's not clear. There is no like a pathway that we can clear associate with this lipid.
Okay, still mysteries to solve. Okay.
Yeah, still it is.
All right, great, thanks. So we've still got some room for more questions. I've actually got a question. So I have a question about this brain penetrance. So the lung versus brain distribution of the pathogen. So understanding why, do you think the reason you're not seeing the brain, any colonies in the brain when you do treatment or when you knock out the SGL1, do you think this is a defect in the ability of the pathogen to move throughout the body? Or is it just that you're killing the pathogen in the lungs, and that essentially, if you just don't have a high enough load, it never crosses the blood-brain barrier?
Well, this could be both of these things. Disease could be dying just after being phagocyted inside the macrophage, and maybe it's not moving or disseminating to another place. It's possible that it's dying, the yeast is dying, or it's possible that the yeast even reach the brain and then die in the brain. So this is not clear for us yet. We still need to investigate this better.
Okay, great, thanks. Julie, you should have the ability to unmute.
Yes, I am able to unmute. What a great talk. I did have a question. I was wondering whether you have any ideas about why the deletion is sensitive to low levels of oxygen, and whether this might help answer Jeremy Thorner's question about what the role of the lipid is in the cells.
Well, we don't have a clear answer for that. We know that to the ergosterol, we have just few clues that we know that ergosterol is needed in the condition of low oxygen in this, but and maybe just blocking this degradation that sterylglucosidase does, lowering the ergosterol availability, it leads the strain more susceptible, but we don't have a clear answer yet for that. It's something very interesting to investigate.
Cool, thanks.
All right, we probably have time for one more question. I'll give it maybe a couple seconds here to see if that'll come through.
Call
Yes.
on Luigi De Luca.
Yes, thank you very much. That was a very interesting presentation. Hello? Yes. So about the function of the ergosterol glycosides, is anything known in mammals whether these glycosides are precursors to vitamin D? As you know, ergosterol is the precursor. So without ergosterol in the skin, you don't make vitamin D. Is there any knowledge about these glycosides as to whether or not they contribute to ergosterol concentration in the skin?
Well, there's ergosterol glucoside is not a glycolipid present in animals, in mammals. So yeah, so what they have is a similar, it's a cholesterol glucoside. So I don't think, yeah, I couldn't relate that with the vitamin.
But ergosterol itself is present because it is the precursor upon which UV light works. So is it that they have not been looked for, or is it that they do not exist really?
Yeah, actually, yeah, maybe they have not looked for the ergosterol glucoside there, but I have not seen any publication showing the presence of ergosterol glucoside, just only other kinds of sterylglucosides, not specifically this one. This one is just associated, they say to be specific of fungal cells, ergosterol glucoside.
Thank you.
Welcome.
All right, perfect. We are exactly on time. So Nivea, thank you very much for a really exciting presentation. It's my pleasure to introduce our next speaker, who's Dr. Shirley Tremel, and I'll introduce Roger, who will give her formal introduction. Thanks, Roger.
Okay, so soluble enzymes like phosphoinositide 3 kinases with substrates or lipids in membrane can be regulated by all kinds of things that happen at the membrane. And structural studies of isolated complexes kind of hint at these things by cryo-EM and X-ray crystallography. But really what we wanted to know for a long time was to be a way to be able to visualize really what's going on with the regulation of membranes. And so after her master's work in Tübingen and here at the Yale Laboratory of Molecular Biology, Shirley Tremel joined the group about 5 years ago in order to partially tackle this problem. And really, as a relentless and amazing PhD student, her work concerned the primordial PI3 kinase known as VPS34. And one of Shirley's many innovations in this work was to use electron cryotomography to visualize VPS34 complexes on lipid vesicles. And with this approach, she could directly visualize the steps involved in catalysis of the membrane and understand how the lipidated Rab5 activates this endosomal VPS34 complex. And really, although, really Shirley's moved on now to other challenges in her postdoctoral work at Germany, I'm going to leave it to her to tell you about this remarkable work and how it unfolded over the last 5 years. Okay, Shirley?
Yes, thank you very much. I'm looking forward to telling you all about my PhD work, although it's been a while, but I'm always happy to come back to VPS34. So I will tell you about the structural basis for the activation of VPS34 kinase by Rab GTPases. And so this work is based on this paper. It was published in March this year. And if you want to read more on this, then feel free to have a look at it. And I want to highlight especially the work of the collaboration with 3 specific people. So Yoe Ohashi is also in the Williams lab, and we've been working closely together over the whole project. And whenever I mention VPS34, basically Yoe and me have been working closely together. Then Dasu Morado has been helping me with the cryo-electron tomography, and I'll show you some very beautiful images he's helped me to acquire and to process. And then Jesse Bertram has been helping me with with adding a new Rab to our project, Rab1, and you will see what I mean by that in a bit. And so basically what my PhD work has been focusing on is VPS34. So VPS34 is actually a lipid kinase. So it uses phosphatidylinositol PI as a substrate to produce PI3P. And probably lots of you know that PI3P is basically a very important signaling lipid in the cell. And VPS34 actually is present in 2 complexes in the cell. And each of the complexes has 4 components. And these 2 complexes still share 3 of them. So you have the kinase here in purple, VPS34, and then auxiliary proteins here in pink, VPS15 in both, and yellow backlin. And then each complex has actually a unique subunit here in orange and green, which makes it specific to their targeting location. And if you summarize basically lots of in vivo data and papers, then you can summarize it to a point where complex 1 is known to be active in autophagy. So it produces the PI3P at the omegasome, which then forms the autophagosome and so on. So in the initiation of autophagy, and complex 2 has been known especially to be present on early endosomes and the production of PI3P there in the endocytic cycle. But what's been basically a puzzle for a long time is why are these complexes, which are very similar, still recruited to different pathways. And so I've been working on an in vitro system. So I took the example of the PI3P production on early endosomes, and this has been known to be coupled to Rab5. And I've been using an in vitro assay using giant unilamellar vesicles. And in these vesicles, we can vary the lipid composition, but we can also include a lipid that has a malamide, and thereby we can couple this GTPase Rab5, which has a naturally occurring cysteine, or which we engineered the cysteine to, also to be coupled to the membrane. And then we add our complex 2, and then we can monitor the reaction, the PI3P production, by a fluorescent marker, which you can see here. And basically what you can see is that with Rab5, we have a big increase in activity of VPS34 complex 2 here in red versus without Rab5 here in black. And if you also want to know more about this in more detail, then our collaborator, Joe Falke, has published an in detail single molecule studies about this, where he basically shows that Rab5 increases the on rate of complex 2, decreases the off rate, and also brings about an allosteric activation of the complex 2. So all in unison basically with our results here. But another interesting point here is that now we can juxtapose it with complex 1, and we can see that complex 1 is activated by Rab5, but just not as much, which was kind of a surprise to us. So we can already see differences in activity of these two complexes. And then if we go to the next target, so this has been introduced by Jesse Bertram. So basically we were interested in PI3P production at the autophagosome, and this has been recently or more and more over the years established or related to Rab1. And one thing that she could actually also see as well is that if she uses a bioID screen, which is where Rabs are recruited to the mitochondria with a with a Pereira ligase, so biotinylation, then she can see that Rab1 specifically interacts with the component of complex 1, so ATG14L, the orange complex unit of complex 1, but actually not with the unique subunit of complex 2, so UVRAG. And that was very interesting to us because, again, it's a difference between the two complexes. And so we wanted to follow this up in our assay. So we used exactly the same setup and used Rab1 here and complex 1. And what we could see as well here is that once we have Rab1 on the membrane, we can see a really drastic increase in activity of complex 1 versus if we don't have any Rab1 on the membrane. And here it was even more interesting or even more drastic is that if we have complex 2 by itself, or complex 2 plus Rab1, then we don't see any activation. So Rab1 seems to be very specific and activator for complex 1, but not for complex 2. And so if we look again then at our map that we've seen before, is that we think that these Rab GTPases are very prominent recruiters of these two complexes to each cellular pathway, and they're a big reason why these complexes are recruited to different pathways in the cellular organism. Then, as a second part of my project, I've been working on a structural part. So we are always interested in the structures of these VPS34 complexes, but I was also specifically interested in the structure of VPS34 in correlation with the lipid membrane. And that's been notoriously difficult to establish, but I try to do it anyway. So I undertook the daunting task of learning electron cryotomography, and thereby the lab of John Briggs has been a big help, and especially Dustin Morado and Lexi Koftun have been helping me acquiring the tomograms and processing them. And so basically electron cryotomography is you freeze proteins, or here I actually use vesicles as well in an ice lab and image them, and then we reconstruct actually a 3D volume, which you can see here. So this is an electron cryotomogram. So what you can see here is an ice, and in ice you have the vesicles, which are actually coated in Rab as well with the malamide lipid. And then these black dots that you can see around here are actually VPS34 complex 2. And so you go from the top to the bottom. And so this is basically the end of the ice layer. And one specific thing that we also tried is that complex 2 actually was a difficult target. So what we used is a part of complex 1. So complex 1 has this specific amphipathic helix here in orange that I depicted. And this is a very good membrane binder. So we made a fusion construct to increase membrane binding of complex 2 to the membrane. And so we acquired about 105 tomograms. And from these tomograms, you get a 3D volume, which you then can average and solve a structure from. And so you can see that here again in the zoom in where you see the black, basically you can maybe also see already some V shape of the complex. And so what I was then able to do is that I solved the 9.8 cryo-electron tomography structure of VPS34 complex 2, which was on Rab5 vesicles. And so what you can see here is the typical V structure of VPS34 complex 2. So we often call one arm the non-catalytic arm. This is this arm here, which has this coil in green and yellow. And then we have a catalytic arm, which is this arm, which has the catalytic active site of VPS34 here in purple. And it has, if you look here as well, then you can see actually we have an extra density here in red, which we allocated to Rab5, the Rab5 density. So this is the low threshold version. And then if you go to a higher threshold, you can actually see the membrane appearing as well. And what you can see specifically, and actually that was a surprise to us, is that although this is the catalytic domain of VPS34, it actually is not in permanent contact with the membrane. Although this is the part which actually should have contact to the substrate PI and then produce PR3P. Instead, we get a membrane anchoring that a permanent membrane anchoring through this domain, the yellow domain here, which is called Becklin. And I will come back to this later. So basically the questions that we wanted to answer is, where does Rab5 bind on VPS34 complex 2? How does the complex bind to the membrane? And how does Rab5 activate the kinase activity of VPS34? And so the first question is, so where does Rab5 bind? So we could see a clear density here, here in the high threshold where we can see the membrane, which was positioned in this kind of cleft here. And if we look at the distance that Rab5 has from the membrane, then it's about 50 angstroms. And so a very specific GTPase characteristic is that it has a C-terminal domain, which is also in cells anchored to lipids. And this hypervariate domain, so HVR, is in Rab5 about 34 amino acids. And so with 50 angstroms, we can easily span this hypervariate region, and then gives also the complex still room to move around. And I will come back to this later. And then if we look in more detail, we can see kind of connections to VPS15 here in pink, and then also VPS34 in purple. And because of course, this is a 9.8 angstrom structure, so any kind of atomic details are not easily seen. So we also undergone a lot of mass spectrometry. So I have to summarize a lot of work in one slide, but basically what I also did in collaboration with the lab from Catherine Lang and Yuri Rapserva is unnatural amino acid cross-linking. So I expressed Rab5 with this unnatural amino acid, and this unnatural amino acid has the capacity to cross-link to other proteins. And when we incubated complex 2 with this Rab5 unnatural amino acid construct, then we could get one single cross-link, which was very satisfying. And this cross-link was VPS34 glutamate 202, which is positioned here in this helix. And but in parallel, we were also doing other work. So we were doing hydrogen deuterium mass spectrometry, which was also done with our mass spec facility, and Yohei has helped me with that as well. And basically HDX, you can see also, you can map protein interaction site, and what we could see through HDX. So when you look at these colored regions, these are indications that proteins are interacting. And if we look at this map where we have the interaction site basically colored, and then look at our cryo-electron tomography structure, you can see basically whether we have similar regions. So there's VPS15 WD40 domain here and here, and then also another domain by VPS15, but then also specifically this helix of VPS34. So we were very confident then in our mapping of Rab5 binding site. And actually, I don't have time to show this here as well, but Rab1 binds to the same side of complex 1 as well. So then the other question as well was, how does complex 2 actually bind to the membrane? So complex 2 is actually a peripheral membrane protein. So it doesn't span the membrane, but it actually has a specific loop, or which is kind of the most prominent membrane binding region, which is from this domain here in yellow, which is called Becklin. So Becklin has this loop here, which has 3 aromatic residues, which are known to be very important for membrane binding and thereby also kinase activity. And in our previous study, we've also mutated these residues and then found that it leaves the complex basically dead. So it's a very important membrane anchor. And if you want to read up on this, and this is the paper by Yohei and me previously. But what you can see is also that it touches the first membrane lipid layer, but doesn't span through the second one. Then one thing that we actually saw, which I thought was very curious, that you can also do classifications on these structures. And we saw that VPS34 actually, or the complex is able to tilt up and down. So it has kind of this tilting motion. So when you can see this membrane anchor is basically stable, and then this part can basically move up and down. So the distance between the, which is actually here, the kinase domain of VPS34 increases more or less. And this tilting motion was actually not influenced by vesicle diameter. So I could see it on variation of vesicle diameters. And I thought this was quite a cool thing to know how actually a protein sits on a membrane or moves on a membrane. Then another thing is how is actually VPS34 activated by Rab5. So one thing that is very specific for VPS34, the complex. So you can see here, this is the yeast complex 2. And also there have been negative stain studies. And what you can see here colored in purple is the kinase domain of VPS34, which is anchored to the rest of the protein by this flexible linker. And because it's only anchored by this flexible linker, it has this ability basically to move around completely. So it doesn't stay in one place. And we've seen that also in other cryo-EM structures, that often you can't capture it because it's just so flexible. And one thing that we could see in our, or actually, so a specific thing about this yeast structure as well, is that it was captured actually in an auto-inhibited state. So this green loop here is the activation loop or the substrate binding loop of VPS34. But in this structure, it was actually captured in an inhibited state because it's basically blocked by this VPS15 N-terminus, this pink loop here. And if we look at our structure that we got from the cryo-electron tomography, we can see that once we add Rab5 and the membrane, then this domain basically flips out. So I'll go back and forth again. And then once this flips out, it's actually free again. So we think that either Rab5 or the membrane or both, or causing basically this allosteric activation by freeing the activation loop, and thereby VPS34 is then in an activated state. And so basically, if we summarize all of this again in a 3-step model, then what we think is happening is that VPS34 complex 2 is in an inactive state. Then it's recruited to membranes by Rab5. Once it's basically sits on the membrane anchored by this Becklin aromatic finger, and by Rab5, it is shifted into an activated state. And then once it's in an activated state, it can produce PI3P from PI. And then also by this membrane anchoring by Becklin, we think it might be able to scoot on the membrane. And this also, this flipping up and down could be kind of an in-between a hopping motion that is known for membrane proteins. Okay, and I think I might be a bit early, but I want to thank a lot of people. So it's been a long project, but I think especially Roger and Yohei in the lab, and of course other people, Glenn, Olga, and Laura, have helped me as well with setting up everything about VPS34, the essays. Then Jesse Bertram brought the Rab1 to our project. The mass spectrometry was helped a lot by Yuri Rab1 and Zhu Cheng and Catherine Lab and Marie van Friesberg. And then especially the tomography has been quite a lot. So John Briggs and Dustin Morano and Alexa Coftun helped me a lot with the processing and the acquisition of the tomograms, and then also our mass spec facility. And with that, I stop, and thank you very much for your attention.
All right, Shirley, thank you very much. We've got a lot of questions coming in here, so I'll make this quick. Ray, you want to unmute yourself?
Yeah, I'm curious if you tried to maybe lock the kinase domain in closer to the vesicle by using an ATP analog or something like that. Can you see, or maybe a better question to be asked would be like, could you see any particles or any images where the VPS34 was close enough to the membrane that it could catalyze?
So I guess it's hard. So I should also say that when I made these samples, I didn't include ATP, because at that point it was actually, I thought about adding ATP, but then the whole timeline would be very complicated. How do I freeze my grids and so on? So I was always interested in what would change if I would have included it, and whether it then might have actually touched the membrane, because then it would have been in full active state. But yeah, from this point on, the one class basically was the closest to the membrane, but there might be also subclasses which I don't catch, because the classifications are also quite hard. Yeah, so that's.
Perfect. Next question is from Jeremy Thorner. Jeremy?
Yeah, one point that wasn't clear to me from the work you presented, although it was beautiful, really elegant as most of the work from Roger's lab is the binding and activation of complex 2 specific to GTP bound Rab5A? Are the switch regions involved in the specific interactions? Does Rab5A GDP not bind?
Yeah, so it's definitely GTP dependent. So I've used the QL mutant in these assays. The QL mutant, I've loaded it either with GTP or GDP. So the QL mutant doesn't hydrolyze either of the nucleotides. So and what we saw is that the activation was GTP dependent, and also the switch regions are included in the binding site. So we could also see an overlay that if we have GDP, that it would basically clash. So it's typically like for all GTPases, this is GTP dependent.
And any similarities to how Reb associates with the TOR kinase as shown by Nikola Pavletich? Were there any homologies or not?
That's a good question. I think I might have to give that one to Roger if he wants to answer that. To be honest, I don't know. That might be a Roger specific question.
All right. Thank you.
Thank you.
All right, Dan Rabin.
I just have one quick question. Jeremy asked my GTP question, so I'm going to ask just a quick one. One of the things I noticed in your structure, which I thought was beautiful, is there was actually a lack of a lot of flexible regions that I've seen in other kinases. And I'm just wondering, in the selection of your particles, was there, did you mask out a lot of the flexible regions? Was there any masking going on in that is a question?
So I didn't mask it. Well, I guess I did. So basically, when I started off choosing particles, my mask was specifically on the bulk region. So it was far away from the membrane, because the membrane basically screwed up any alignment. But I'm not sure if you can see my pointer, but it was basically on the top of the protein. But that would mean also the kinase domain was kind of out of that mask, so it wasn't technically included in all of the, depending on the threshold. And I at some point I also chose specifically particles which had the VPS34 kinase domain present, just so that I can basically focus on that. But it's hard to tell whether the rest of it was basically noise that kind of aligned in the VPS34-ish shape, or whether these were actually particles which didn't have the kinase domain structured, it's always kind of hard to tell from classes at this point.
Sure.
So I don't think it could be still a possibility that there's still some classes which don't have the kinase domain structured. I see what I'm saying.
Sure. And just one very quick question, if I may. Have you thought about trying to model in a lipid substrate? I'm kind of curious about using your structure to model in how you would affect the off rate of the Rab proteins.
Yeah. Yeah. I mean, so the 9.8 angstroms doesn't give a lot of room for. So we can shift domains and so on, but unfortunately, this would have to go far higher. And I'm not sure how much tomography you would have to do to get to that.
Well below 6. Yeah.
But it would be interesting to know that the single molecule studies by Joe Faki were like, that's a really good paper to read up on that as well, if you want the kinetics.
Beautiful work.
Thank you.
All right, Kusum Karbanda.
Thank you for taking my question. So are these Rabs methylated, and does that affect their membrane association?
So they have a geranyl lipid anchor. So basically there's an enzyme, geranyl transferase in the cell that transfers 2 lipids to cysteines, which are in this hypervarial long region. So I guess they're lipidated, if that's what you mean.
No, actually, I'm asking. They have to be lipidated and then they are methylated, and that kind of traffics them to the right place. I think association with the membrane, and that's something that most of the GTPases have. But I didn't know if the Rabs have the same kind of structural modification, the post-translational modification that will allow it to be methylated, and how that will, and whether these Rabs are methylated.
I'm not sure if I know exactly what you mean by methylated.
So the cysteine residue is methylated by the ICMT methyl. That's a methyl transferase that kind of donates some methyl group from acetylmethionine to these GTPases, and that's what kind of determines their activation and association with membrane, actually. So that's what my question was, whether that would affect the membrane association also.
To be fair, I'm not a Rab expert. I'm sure there's lots of things. I don't think it's been like properly determined what really affects the membrane association and why one Rab goes to another membrane versus another Rab goes to a different membrane. I might be not the right person to ask this question. I'm sorry.
That's all right. Thank you.
All right, we've got another question from Alejandro Alvarez Prats.
Hello, beautiful work. Thank you so much for that presentation. So just from your videos, what's my impression that there is like kind of tubulation formed between the vesicles. And I was wondering if that's communication between the vesicles, maybe to share these BPS34, or what are your thoughts on that?
Yeah, so I think these might be tubulations which are actually caused by how these vesicles are made. So you do a lot of freeze-thaw cycles and sonication. And I think that's more what it looks like that there's tubulation. I don't think, if I look at the vesicles by themselves, they look identical without and with VPS34. So I think the tubulation is not something that is caused by VPS34, but that's just caused by the production of the vesicles. Although it would have been really interesting to see that. And so we had this thought as well. But at this point, we don't think that there's kind of a membrane deformation that's caused by VPS34.
Thank you. Great.
Excellent. Looks like Scott has just a comment. Scott, do you want to say that, or do you just want to, I can read it off. Scott says a beautiful work. He's not unmuting. So I actually have one final question, since we still have a couple of 1 minute here. So Shirley, very beautiful. my main question is it has to do with this Rab5, Rab1 specificity. So Rab5 can hit both complexes, although not as effectively at complex 1, where Rab1 really seems to be much more selective. But the binding site is going to be present in both complexes because it's not made up of the unique subunit. So my long-winded intro to the question is, do you think this is based on the linker length of the tail of the Rab, so that it's actually an orientation issue of the complex? Or is it a conformational change induced by the accessory subunit?
I think it looks like there's a lot of room for the linker to span a lot of distances. So I don't think it's something that the linker limits. Although we don't have like super high resolution structures of the human complexes, there's a structure of the complex 1, of course. I think I can't remember what resolution, but you can see the coil of the adapter arm, and basically the coil is slightly shifted to another side, and because I guess the coil also guides the VPS34 how it sits, like this helix that it contacts. I think it just shifts the residues that Rab5 can basically, or Rab1 then can interact with, and thereby it's shifted. But yeah, this is a question I wanted to answer at the end of my PhD, but I didn't have enough time to. We made some mutants, but we saw that also these two, basically we made a mutation in VPS34 in this helix, and that Rab1 and Rab5 reacted differently to them. So we think it's really the specific interactions that are guided by the differently oriented adapter arm, this coil arm.
Great. Does Rab1 have Rab1 has a slightly shorter HVR, right, than Rab5, but it's not that much shorter. I think it's like 20 amino acids, but probably still accommodatable in that 50 angstroms that you're seeing.
Yeah, I definitely looked it up, but I don't know the amino acid length at this moment.
Okay.
Yeah.
Great. Looks like we've got one final question. Nancy Hart.
Yes, I'm curious if you've explored how varying membrane lipid composition, such as varying cholesterol content, might affect binding and enzyme activity, not specifically in the size of the vesicle, but just the basic composition.
Yeah, so basically we did a whole lot of studies, and this is actually the paper that was published before. So that's the paper here in the corner, where we varied the lipid saturation. We also included cholesterol. And basically what we saw is if we make the membrane more rigid, so we have more saturated, then VPS34 doesn't bind as well, and then activity goes down. But if we make the membrane looser, so more unsaturated, then we increase binding and activity. So we saw a clear correlation with that, with the lipid saturation.
Thank you.
All right, with that, I think we've had all of our questions. Thank you again to both Nivea and Shirley for two really excellent talks. As a reminder, the next talk will not be happening in October. It's going to be happening the first week in November, November 3rd. So keep that in your calendars. We look forward to seeing all of you then, and hope all of you have a great month.